Okay, so this slide tells you the success of vaccines. So if you look at this slide, it tells you that following vaccination, when we got to 1980, we eradicated smallpox because you can actually immunize and then get rid of the infection. And this is again to show you three different diseases: diphtheria, polio, and measles. And it shows that when you introduce the vaccine, there is a precipitous drop in the incidence of infections, whether it be diphtheria or polio, and even measles. And in measles, you get it as a child, and then you can get it again, and it comes back in SSPE. And then what this shows is once the vaccine was introduced, the incidence of this SSPE also goes down precipitously. Okay, and so you want to get vaccinated, and you do not wish to be an anti-vaccine. Okay, so what is the purpose of vaccination? It does three things. One, it induces memory B cells so that you have an army of cells that can recognize the pathogen that you've seen before. You have memory T cells, CD4 and CD8 T cells, that can recognize previous insults. And also, you have helper T cells that will help memory B cells to give you a profound immune response when the antigen comes a second time. And better yet, you have this creation of plasma cells that can produce antibodies for a lifetime, and they reside in your bone marrow. So if I had chickenpox when I was 5 years old, and I'm 50 years old, for the last 45 years, I've been making a lot of these antibodies because I have these plasma cells that continuously make these antibodies. Okay. Right. So now, once you have vaccinated, what happens when you get exposed to the pathogen again? Step number one, you have circulating antibodies that will form immune complex with the pathogen. You activate the complement pathway. You have inflammation. You have activation of memory CD4, CD8 T cells. You have enhanced killing of the virus infected target cells. You have activation of memory B cells and the different rapid differentiation to plasma cells. And you have massive antibody production. And for the most part, you do not even know that you got a secondary exposure because the system is so efficient at dealing with repeat responses to things seen in the past or things that you have been vaccinated to. Okay? And finally, what that does is the pathogen has no chance; it is decimated. So what the pathogens do is to escape this, they mutate away and they change, and that becomes a challenge. Like in flu, that you vaccinate, then you control the virus, and then it mutates, and then you make another vaccine and so forth every year. So this is an introductory slide. There are three stages to host defense. The first is innate defense. Innate defense, it's like the barriers that you have, like the Great Wall of China, the barbed wire, the things that separate you. And then you have adaptive immunity, T cell and B cell response. And finally, you remember things. It's like the serial, the soap operas you find on television. The Sas and the Bahu stories. They always remember everything. Have incredible memory of all the past insults. And so this shows you a duration of infection and the level of microorganism. So the level of organism, it comes in, it divides, it gets to a high number, and then you need to knock that down to a threshold level by the innate immune responses. Only then can your adaptive immune system start. Okay. And if you are missing the innate immune response, then if you see this line that goes up, the pathogen number goes up, 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 and through the roof, and then you cannot get over it, and then you most of the time you die. I have to tell you, all of the adaptive immune system comes into play, and it's crucial for sure. But if you have no innate immunity, you could be dead as a doornail. So let's move to B cells. So you have seen this 1,000 times. I'll just go through it very quickly. You have a large repertoire of B cells, and each B cell encodes a single specificity for the most part. Okay? There could be some level of promiscuity, but for this lecture, let's just assume that it's monospecific. That's because it has only one specific rearrangement in the like Shiv referred to. And so when antigen comes in, it picks out from this large lineup of B cells those that are specific, and they are induced to proliferate. And then you have all the subsequent reactions that go on that I will talk to you in the next 20 minutes or so. Okay. So we need to define a few things. We need to talk about what antigens are, especially what an antigenic determinant or an epitope is, and then we'll talk about affinity. We'll refer antibody, we'll refer to affinity and avidity. So shown on here is this is the antibody combining. So this is the part of the antibody that binds to the antigen shown on this blue, and the antibody does not swallow the whole antibody. It only sees a small piece of it. Okay, and that small piece that it sees, it's called an antigenic determinant or an epitope. And so what happens is you need to have a perfect fit in order for it to bind really well. So this is a good fit, and the antigen antibody complex stays bound. And on the right hand side, this is a poor fit. This antibody can bind to it, but it is not perfect in its binding. So what happens? This is a low affinity interactions. And the reason why you need an excellent fit is because the interactions that hold the antigen antibody complex together are actually very weak interactions, including hydrogen bonds, ionic bonds, hydrophobic interactions, and van der Waals forces. So what that means is these interactions operate over a very small distance. And so you need to have like a lot of these together in order to hold it tight. It's like a Velcro. Having one of them is not good enough. When you have many of them, it's almost impossible to pull them apart. It's the same thing with antibodies. Okay? And this is just to tell you that you can actually measure the strength of this binding in terms of affinity and also avidity. And people tend to use this interchangeably, but it is not. Affinity just tells you one combining site and one epitope. The strength of binding, that is the affinity. And the avidity is the sum of all the if you have like an IgM molecule that has like 10 combining sites, if you added all of them, you would call that the avidity, right? Very good. So the antibody avidity is based upon the isotype because you can have either 2 combining sites or 10. And so the avidity is the total strength of interaction between multivalent antigen and the antibody. Okay? Right. So I'll just summarize the effective functions of antibodies in just one slide. So B cells get activated, you make antibodies. These antibodies can neutralize microbes and toxins. They can opsonize and phagocytose microbes. Okay? So they bind to the microbe, and the Fc receptor of this antibody that's bound to the microbe can bind to an Fc receptor on a phagocyte and can be taken in easily. And also, like Shiv referred to, this antigen antibody complexes once formed, they can activate the classical pathway of complement, and which leads to lysis of the microbe, phagocytosis, and inflammation. Okay. All right, very good. So cross-reactivity. In the first slide, I showed you that the B cells are very specific. But however, what happens is, even though they are specific, the antibodies can cross-react with other related antigens. So what happens is, when 2 different antigens share a similar epitope, and this antibody cannot discriminate between the two. Okay. So when you make antibodies, and what the bug does is the bug keeps changing itself. And it wants to have the epitope look like some part of your body, so that the target for this antibody is much more to attack your organs in your body than it is to attack the microbes. And here are a few examples. So molecular mimicry is when the epitopes between the pathogen and the humans look very similar, and the antibodies made against the pathogen can cross-react with the self-antigens and cause disease. And this is just there are 3 categories. So the exogenous antigen, Streptococcus M protein, it looks like cardiac myosin. So when a child gets infected with Streptococcus, you make the antibody, and there's much more cardiac myosin than there are bacteria, and then the antigen will attack the cardiac myosin and cause rheumatic fever. Similarly, Yersinia protein can cause Graves' disease by cross-reactivity. Papilloma E2 looks very similar to insulin receptor. Measles can lead to EAE. MTB can cause rheumatoid arthritis. Poliovirus VP2 looks like ACH receptor, and antibodies can cause myasthenia gravis. And the bacterial phospholipid looks like DNA and can cause SLE. Okay. Good. So 2 things to remember: polyclonal versus monoclonal. Polyclonal means that I told you one B cell can make one specificity. It attaches to one antigenic determinant. But as shown here, it's a bacterium. It has many different epitopes shown by all these sizes, and you can make antibodies against all of these different epitopes. So the sum, all of these antibodies, that'll be a polyclonal response. But if you can isolate just this one clone that makes antibodies against this one determinant, that is called a monoclonal. And the people that would discover this would go on to win the Nobel Prize. Okay, why is this important? This is important because this has actually changed the way treatments have evolved. It started out with murine monoclonal antibodies. You would make monoclonal antibodies, inject into humans, and you could cure diseases. But the problem was you would make a human anti-murine antibody response, and that was not good. Then what they made chimeric antibodies, where 30% of the chimera is murine and 70% is human. That is actually pretty good, but nonetheless, it's still you can trace antibodies against the 30%. And then came the humanized antibody, where they take the murine or rodent antibody, they keep everything except the antigen contacting sites. And so the CDRs are rodent, everything else is human, and that was great. That worked beautifully. And the last type, what we do now, is human monoclonal antibodies. So you just sort out the B cells, clone out the heavy and light chain, you can make the antibodies, and then you are in business. Okay. So there are 4 types of therapeutic monoclonal antibody. One is the mouse, pure mouse, and you can tell it by the way that it is named. The name ends in MOMAB, so it's a mouse antibody. Chimera would be CMAB, like rituximab. And then if it's humanized, it's actually a rodent one, but the rodent part is just the top part shown in blue. It's a Zumab. And if it's completely human antibody, it's called a MOMAB. So next time you find an advertisement on television or on a journal, you can actually tell what kind of antibody is based upon the last, how the name actually ends. Okay. So I'll just give you like 2 examples, and then I'm going to move on. So rituximab is a monoclonal antibody directed against CD20 on B cells. It's beautiful because if you give it, it can actually deplete B cells. And this is a patient that came into the clinic at Emory, and he has pemphigus, and which he's an autoantibody disease, and they bring him in, and then they treat him for 2 months versus 4 months with the rituximab, and then lo and behold, in 2 months, all his lesions heal up because it kills it depletes the B cells, and then it's completely cured. Okay, and if you look at turn on television in the United States, you see this commercial for Humira all the time. It is actually a recombinant IgG1 monoclonal antibody against TNF alpha. This is actually the top-selling drug in the United States today, about $17 billion every year. And if you look at what is the drug that makes the most money in the US, right? The 7 out of 10 are actually antibodies. So the point I'm trying to make is the applications of this are actually super, super powerful. So there are 2 types of active immunity versus passive immunity. So in active immunity, you induce immunity in the host by activating the immune system and generating immune memory. You do that by either getting infected or by vaccinating the person. Okay, and usually you have to vaccinate before the anticipated exposure. Right? If I'm going from here to, let's say, Africa, and I'm worried about yellow fever, I should get the vaccine now, wait a month or so to get the antibody responses going and the T cell response, and then go to the spot. Right? The exception is rabies. You can actually wait for the dog to bite you, and then you can actually go get the vaccine. And that is because the incubation period of rabies is much longer than it takes for your immune response to develop. So that's okay. You don't have to, everybody go get a rabies vaccine and hopefully you get bitten. You don't have to do that. You can actually do it post-exposure. Okay? And passive immunity is the transfer, and we hear this a lot now. It is the transfer of convalescent sera monoclonal antibodies that you put into a patient, and then you can actually cure them by doing this. Convalescent sera used to be what they used to in the old days, but now we move on to monoclonal antibodies. And these things are actually very efficient. So in 1988, serum from horses were used to immunize. Serum from horses immunized with diphtheria toxin were used to treat patients. Okay, 1890, we developed tetanus anti serum. But the problem is you can only do this once, because if you do this again, then you make an anti-horse response, and then you know what happens next. And if you got botulism today, you still get a horse antitoxin, and hopefully you don't get botulism a second time. And we already know from the Ebola outbreak and also this current COVID-19 you can actually transfer convalescent plasma, and then you can actually contain or control the infection. And I have to remind you, during the outbreak of Ebola, this is my university, Emory University School of Medicine in Atlanta, and this is the missionary doctor that got infected um, with Ebola, and he came to the U.S. and they gave him a monoclonal antibody. I'll show you in the next slide. And he's cured, and he's leaving the hospital. He's very happy. And so what they gave him was ZMapp. They have a 3 monoclonal antibody cocktail that they made in transgenic tobacco plants, and they give him the antibody, and then they can cure him. The thing you have to remember is you need to put like it's not a simple exercise. You had to put a lot of protein into a person. So the scale or the magnitude of antibodies that you require is actually quite high. And so then this is a doctor that got cured, and this was a nurse in Dallas that got infected. So he cured her by giving his convalescent plasma into her, and she gets cured. And this is the gentleman that died, and he's from Liberia. He died in Dallas, and you could not give a plasma transfer to him because there was a blood group mismatch. So you cannot you have to give plasma only if you're of the same blood type, and no incompatibilities. This one worked out, this one did not work out because of the incompatibility. So that's why now we make monoclonal antibodies, and that you can give it to anybody, and it works really well. And this is in the news. Regeneron is a company in the States. They make these antibody against the SARS coronavirus, against the spike protein. And you can actually give the antibody, and the hope is that you can actually cure people. And they actually gave it to our fearless leader, and this is the antibody that the President Trump received like last week. And the outcome, it's not published yet, but the hope is that it's excellent therapy should have taken care of his viremia, at least to some extent. So now we have talked about, just to recap, antibodies are important. They are important for serological memory. I talked about one lymphocyte, one specificity. Then we talked about monoclonal antibodies, the applications, why this is so important. And now we'll just get the nuts and bolts of what happens when a B cell gets activated, and what do you do? How do they go on to establish memory? And this is not, and this topic, when we teach it at Emory, this is like a 3-week class where we go into each individual thing. But this is a quick review of what happens. It's like a 300-feet view of it. Okay. So antigens comes in 2 flavors: T independent, which, as the name suggests, you can activate a B cell without T cell help, and obviously you have T dependent antigens which require T cell help. And in a T independent antigen, you have the B cell receptor, B cell, and you have B cell receptor. And what happens is the antigen can activate the B cells in the absence of T cell help and make them differentiate into short-lived plasma cell. And when you do this, what happens is that there is no immune memory for the most part. You need T cell help in order to establish immune memory. So when your nephew or niece or your child gets repeated ear infections with bacteria, that's because it's a T-independent response. And so they can infect you again and again and again and again. If you have a T-dependent response, you will have this part of the response. Plus, you'll open up and have a much more elaborate immune response. So if it's a T-dependent antigen, you do make the short-lived plasma cell response. And you also make a T-dependent germinal center B cell response. And in the germinal center B cell response, this B cell undergoes mutation and selection. And then the B cells that can have the most high affinity or better binding antibodies survive. The rest of them perish. And those with that jackpot mutations or high affinity go on to become plasmablasts. They go to the bone marrow, and they live in the bone marrow for extended periods of time, and they make antibodies for their life. They can make between 10 to the 4th and 10 to the 5th antibody molecules per second. So it's a factor. You don't need a whole lot of these cells to make a lot of antibody. And I have to tell you, based upon the isotype, if it's IgG, 2A or something, you have 21-day half-life. So it's very easy that you make a high amount of antibodies in your body. And from the germinal center also comes memory B cells, high affinity memory B cells that stay and they wait for the antigen to come again so that they can actually differentiate faster and go on and become plasma cell, make more antibodies. Okay. So T-dependent antigens, these are the characteristic T-independent antigens that are type 1 and type 2. And I just put the slide. You can look at it at your leisure. I'm going to skip in the interest of time. So there are two events that happen during B cell differentiation. One is class switch recombination, and the other is affinity maturation. Okay, class switch recombination. So this is a antibody. Everybody starts off as IgM, and when this B cell gets activated, it keeps the specificity the same, the front end the same, and it switches out the rear end for another heavy chain isotype. So you go from IgM to IgG. The front end remains the same. The back end changes. It's like one of these, 18-wheeler trucks that you see on the highway. So the front end is always the same. And today they'll be carrying, produce for a grocery store, and tomorrow they could be carrying, gasoline for Indian Oil or something like that. So the front end is the same, and the back is the one that you keep changing. And the reason why you do this is because you can actually fundamentally change the effective function of antibodies by doing this. Just to quickly, IgG, D, and E have 2 combining sites. IgA has 4, IgM has 10. That's one thing. So the avidity, like I told you, avidity would be so if it's the same combining site, right? The affinity of the IgG and the IgM is exactly the same because it's one combining site and one antigenic determinant. But if you look at avidity, this is 2 and this is 10. So you have 5 times more combining sites. So the binding efficiency, avidity, is significantly higher because of the valency of this antibody molecule. And the reason why you switch between these is so this is the rearranged VDJ, this is the C mu, and all the downstream switch heavy chain isotypes. So what happens is you have these switch regions, S mu, and other upstream of each of these heavy chain isotypes, you have a switch region. And you switch between them. You bring them together, you cut it, and you glue it together. And now you went from an IgM to IgG3C gamma 3. If you switch all the way to the end, C alpha, now you have an IgA antibodies. And once you recombine it, you cannot say, oh, I just changed my mind. I want to go back to be an IgM. That is impossible because you have actually deleted the intervening DNA at the genome level. So what controls which way the B cell decides to switch? And it turns out it has to do with the cytokine that's produced. So the T cells that make the cytokine, during the immune response, the cytokines that are produced will actually determine what isotype of B cell switches to. If you make a lot of IL-4, go to IgG1 or IgE. If you make a lot of TGF-beta, which is what you find in the mucosa and in the gut, you have a lot of IgA that is induced. And depending upon the isotype that you switch to, you have a lot of these different functions that it's the same antibody, but by changing the rear end, you have completely changed the way that the effector mechanisms that are involved in this antibody. I just show you just a couple. You don't need to go. You have seen this before if you have taken a class. If you look at complement activation, there are some that do, others that don't. If you look at placental transfer, there's some that do, most of them that don't. If you look at the binding of macrophages and phagocytes, some do, the others don't. If you look at binding to mast cells and causing degranulation, all the IgE do, and so forth. Okay, so there is a consequence to switching your heavy chain isotype. But the most important thing is the isotype changes. If you want to make a vaccine and you want it to be a long-lived antibody response, you want to hit it with an adjuvant. You put the right kind of adjuvant that makes the right kind of cytokine that forces the B cell to switch to the isotype of your choice. So you want to hit something that goes to 20 or 21 days as opposed to something that goes like 3 days or something. You want to stay away from things that you vaccinate and you make an IG response because you don't want to do that at all. It's not going to end well. All right. So now we'll get into mechanisms of, how do you bring about these class switch recombination. Okay, so this is Dr. Honcho. He won the Nobel Prize. he discovered an enzyme called activation-induced deaminase, AID. And this enzyme is important because it induces class switching and immunoglobulin um, somatic hypermutation, which I'll tell you in a minute. Okay. All right. So in class switching, what happens is again, so this is the VDJ, and you have the switch sites ahead of everything. So what the AID does, it makes a cut, and then you glue the cuts together by non-homologous end joining. So you have AID-mediated double-stranded break. You break it, and then you have end joining, and then and that's how you bring these two pieces together, and the intervening piece gets thrown out. Okay. This is just to show you that it's the same slide as before. You have VDJ, C mu, and C epsilon, and you have AID that cuts, and then you glue it together, and now you have a VDJ C epsilon. You made an IgE antibody. All right. So B cell differentiation. So a naive B cell gets activated. You have membrane-bound IgG and becomes plasma cell. It secretes the immunoglobulin, and the way that you go from membrane-bound to secreted, you do it by differential splicing, and everybody knows that. I just put it for the sake of completion. And the other thing that happens is affinity maturation. So when you start out, the affinity is kind of low. With time, you make better and better higher affinity antibodies, and this progressive increase in affinity or affinity maturation is caused by two events. One, you mutate, and then you select for the high affinity clones. Okay, all right. So this is a busy slide, but it's actually very not that difficult. So this just shows you day 7 of an immune response, day 14, and day 21. This is heavy chain and light chain. Let's not worry about the light chain. Let's just do heavy chain. If you look at mutations, you find that there are mutations that are introduced into the heavy chain. And what happens is with time, the mutations get selected in the CDR regions. And that CDRs are the ones that actually contact the antigen. And by selecting for mutations that are in the CDRs, you start out like it's a poor fit. It binds the combining site, that's the epitope. Then you mutate, and then now it becomes a perfect binding and a high affinity binding, and then that is very stable. So there are two things. You make a lot of these mutants, and then you select for the ones that actually bind the best. And the rest of them that do not bind are not enticed to enter into the next level of selection. Okay? So antigen-driven B cell differentiation. So this is just a stepwise progression of the things that happen. Day 0, antigen comes in, the DC state. So again, this is the T cell zone, B cell zone. This is the interfollicular region. So the dendritic cells take up the antigen, they present it, and those T cells that can see the MHC plus antigen will come and get activated. Similarly, the antigen in the B cell zone, the B cells that are specific for the antigen will bind to it, and then they get activated. And once they get activated, they leave their homes, right, from the T cell follicle and from the B cell follicle. They come outside, and then the B and the T cell stay glued to each other, and it's almost like they are kissing, and they do that for almost 2 days, right? It's almost like a newly married Indian couple on their honeymoon. And then what happens is on day 4, 5, 6, and 7, they come back into the B cell zone, both the B and the T cells, and now they start expanding and they push everything out. And then now you have what's called a germinal center that is formed. And it is in this germinal center that you have this affinity maturation that happens. Okay? All right. So this is a germinal center, like I showed in the previous slide, that you have antigen that's bound to the follicular dendritic cells. And first is seeding. The B cells and B and T cells enter. In the second, you have proliferation and somatic mutation in the dark zone. And then they move up into the light zone where they are selected for by binding to the antigen. If the B cell mutated away and cannot bind the antigen again, then it stops there. It cannot go forward because you need antigen binding to rescue these cells. And then you go through cycles of these, and at the end of it, what happens is you have those B cells that bind very well, high affinity ones that can go on to become long-lived plasma cells and memory B cells, right? This is sort of like what happens to, those of us who are faculty members. Everybody enters, right, with such promise, and we're going to save the world. And then we go there, we mutate, and then as life goes on, we get selected and selected. Very rare of us make it. Most of us don't, and that is life. It's the same thing in a Germinal center. It's just reflection of life itself. And the Germinal center, this I got this from I think one of Jason's sister's slide. The Germinal center is a microcosm of Darwinian evolution, and it's evolution steroids. Okay, so in the dog zone, they proliferate, they come back in again, and they get selected, they come back in, they make more of it, they get mutated again, and you have this multiple rounds going, and at the end of it, you have really high affinity B cells that are made, and you have this selection, and it works because you mutate. That's good, but the selection is what makes this very, very efficient. Okay. And now we'll talk about antigen-driven B cell activation, generation of B cell, and serological memory. Okay, so this is like a summary slide that I like to show you. So a naive B cell gets activated, you become short-lived plasma cells, which make antibodies in the short term. That lasts very short time. And this part happens both in T-independent and T-dependent antigen. If it's a T-dependent antigen, then you have this plus this Germinal center reaction, where this B cell, purple cell, like the color of my shirt, comes in, and now it undergoes somatic mutation. We make all these different colors. And the red one, that's a high affinity clone, is selected to become enticed to enter the memory B cell pool and into this long-lived plasma cell pool that they become plasmablasts. They go to the bone marrow and they take residence, and they make antibodies for a long, long time. Okay, and so the question is, why do these plasma cells in the bone marrow live for a long time? The reason is because in the bone marrow there are these cells, these stromal cells, that hold this B cell, this plasma cell very carefully. And if this stromal cell lets go of the plasma cell, then that plasma cell dies. Okay, because they have shown that you can actually take antibodies against BLA4 and VCAM1 and so forth. And if you interrupt this, then the long-lived plasma cells will actually die out. And you also need like other cell types that like eosinophils that make APRIL. These are like growth factors that are required for the plasma cells, and also for an IL-6, which is required for them to survive. So they need to be held and they need to be fed. Okay, and if you let them go and they just crumble and they die, they're like some people that you and I know in our families, right? You have to be very careful. They're so touchy-feely that they had to be held carefully all the time. One small thing and the whole world crumbles. It's the same thing with this long-lived plasma cell. And in fact, if you actually make this is APRIL, if you make a BCMA FC, that is, if you mop up all the APRIL that's made, then it dies also. It dies of starvation. Very good. So again, the long-lived plasma cells live because they can bind to they form interaction with the stromal cells, and there are other accessory cells that meet their needs by giving them the growth factors that they require for survival. And in return, what the cell does is it makes antibodies for a lifetime, like 10 to the 4 to 10 to the 5th antibody molecules per second. And so what happens then? So that's one half of it. So you establish your long-lived plasma cells. I also said you make memory B cells. Those memory B cells go, and now they become part of your primary repertoire. And when this antigen comes again, oh my goodness, these memory B cells are like pumped up and ready to go. And they make a massive proliferation. They get so much plasma cell, and they make so much antibodies that you don't even, and it decimates the pathogen so efficiently that you don't even know that you had actually a clinical manifestation, because your immune response took care of it exceedingly well. Okay, so this is the slide that you have seen in Immunology 101. This is antibody concentration in the serum. This is time. You vaccinate or you first exposure, you get a modest immune response, and this takes about 7 days. The lag is actually pretty long. You get a secondary antigen. The same antigen comes back, and then lo and behold, you get like 2 to 3 orders of magnitude. You make much more antibodies, and you could make it much more faster than you did in the primary immune response. So when you vaccinate, what you do is you are priming the system to make memory B cells and serological memory, so that when the antigen comes again, the antibody that's existing will bind to the antigen, make an immune complex, and then the T cell memory CD4 T cells will have the memory B cells give massive activation and plasma cell generation and 2 to 3 logs greater antibody production in the serum. Okay. And so if you compare primary versus secondary, you've seen this a lot of, you have the primary response takes 4 to 7 days, secondary is 1 to 3 days. The peak response is about 7 to 10, this is 3 to 5. And if you look at affinity, the primary is lower and the secondary is usually higher because you get them from the pool of your memory B cells. Okay. So this is my last slide. So why are memory B cells better than primary B cells? I used to have like another 10 slides that I go through experimental data, which I decided to take that out. It's because of two things. Number one, the memory B, if you look at, so they did this an experiment where they took a B cell receptor from a memory B cell, and then they made a mouse by, the way they made Dolly the sheep. They transferred the BCR into a new animal so that every B cell has this memory B cell receptor, highly mutated one. And they wanted to know if they immunized, did they get a secondary response or not? The answer is no, because the memory B cells work because they are epigenetically and transcriptionally distinct from the naive B cell so that they can actually respond much faster. They can differentiate faster. They have all the things ready to go and they're wired to respond much faster. Okay. And most importantly, if you need helper T cells, memory helper T cells, in order for a memory B cell response to work. If you don't, experimentally, if you don't transfer in memory B and memory T cells into a new animal and immunize, if you only put memory B, you do not get a massive second response like you do with the when adding the combination. Also, immune complexes are very important. You can actually make an antigen antibody immune complex just by itself and vaccinate and get like a massive immune response as well into a naive animal. And with that, I'm going to stop, and I'm happy to answer questions.